Okay. So the data that we're working with here is collected for a disease called bacterial spot. It's caused by 4 different species of Xanthomonas. You can see the leaf symptoms, and this can lead to essentially defoliation, which also leads to a lot of quality issues. Reduced photosynthetic capacity reduces yield. Fruit get exposed to the sun, so they get sunburned. On top of that, you can get these huge lesions that make the fruit completely unmarketable. Okay, so it's an economic trait. The data that we're going to be dealing with are scored on a Horsfall-Barrett scale. So it's a quantitative scale. If you can look up here what that scale actually involves. When we analyze our Horsfall-Barrett data, the distribution usually approaches something that looks normal. So ANOVA and regression approaches are appropriate. In contrast, if you were scoring this in the greenhouse, we would be injecting bacteria directly into a leaf, and we would be scoring it on a non-parametric scale. Either we get a hypersensitive reaction, which is a resistant reaction, shown here, or we get no reaction or water-soaked lesion, which is a susceptible reaction, shown here. Okay. The data that we're dealing with is on this scale. The point that I want to make here gets at the basis of the population. So over time, we not only identified but mapped resistance from several different sources. In order to actually breed a resistant variety, we need to figure out how to combine that. So most of our mapping was done using this advanced backcross and inbred backcross approach. What I'm really emphasizing with this from a breeding standpoint is that these backcross-based approaches allow us to integress genes from unadapted or wild species into an elite background. So we've got, out of these populations, we have lines and even more advanced material that you would say look good, that is, they're resistant and they have yield and quality characteristics. But we need to combine this resistance. Okay? And so in order to do that, we develop a population where we create a bunch of hybrids, and then we begin to intermate these hybrids. So these are our F1s here, and these are intermated F1s. And so these progeny are beginning to segregate. The crosses that weren't made here were not made to eliminate fresh market material from the background. We take this large population, we go to the field, and we select only the extremes. Okay? This material then gets advanced to the next generation where we can replicate the trials. Okay? And so I'm going to focus on the T1 inoculation and this population of 110 individuals. Okay? We made selections in this year 1 that's single plant. We ask how well does the resistant or susceptible or the Horsfall-Barrett score predict performance in the next year. It's highly significant. We can use that for parent-offspring regression to look at our heritability, 0.32. Okay? Plants rated as resistant in year 1 produce plots with an average disease rating of 4 in year 2, whereas those rated as susceptible, it's a full point above on that Horsfall-Barrett scale. Okay, and our realized gain in that 1 year is a 13% decrease in disease.